welcome to lab number 2. Lab number 2 is all about histology, okay? We defined histology a while back in the first lecture. Histology, of course, is the study of tissues. And this is what we're doing today. We're going to look at a tissue. The title of today's lab is Epithelial Tissues, Simple and Pseudo-Stratified Epithelia. Now, I want to begin by just reminding you that a tissue is a group of cells with a common structure, common function. And there are 4 basic general types of tissues. There's epithelial, connective, muscle, and nervous. And remember, epithelial is your covering and lining. Connective is going to bind and support. Muscle is all about movement. Nervous is all about control. Now, today, we are looking at epithelial tissues. Lab number 3 is epithelial tissues. Lab number 4 is epithelial tissues. 5 and 6 are connective tissues. Then later on in the semester, we'll hit muscle, we'll hit nervous. So if you look over here, this is a microscopic picture, okay? This is tissue from the kidney. There are a lot of cells here. How do I know there are a lot of cells? Well, I can see all of these purple nuclei. All of these purple circles are nuclei. If I have nuclei, I've got cells. So these are all nuclei, these are all cells. On some of the cells, I can kind of see the outline of the cell. Look at this guy right here. His neighbor next to him. On some of them, it's a little bit harder. But I know that I've got cells here because I'm seeing these purple nuclei. I'm seeing the light pink cytoplasm around them. So this is some epithelial tissue. And we'll look more at this exact epithelial tissue in a bit. Now, remember, epithelial tissues, well, what do? They are going to cover surfaces, internal and external surfaces, covering and lining. That is what I was referring, that was the definition, there we go, that I gave you in lecture. Now, every time you have an epithelium, there is going to be some sort of neighboring space. So this right here where this yellow bracket is, this is an epithelium right here. So there's space next to it. This like purplish line that the red arrows are pointing at, that's an epithelium. There's space next to it. So there is always a space next to an epithelium. Now, the part of the epithelium that is adjacent to that space, is exposed to that space, is called the apical surface. So my red arrows here, my red arrows down here as well, are pointing at the apical surface of an epithelium. So this is my epithelium right here, and then there's a space next to it, and the surface next to the space is called the apical surface. Now, down here, okay, here's my epithelium, here's the space, so this very edge of that purpleness right there would be the apical surface. Okay? The view that we have, or the image we have at the top is the skin. The yellow bracket is showing me the extent of the epithelium. This epithelium is kind of thick. Look how many purple nuclei we have. Lots of cells. Why would you want your skin epithelium to be kind of thick? Why would you want it to have many layers? Well, the answer, of course, is for protection. Now, in this epithelium, we have a side exposed to the outside space. That's the apical surface. And we also have a deeper side, the one my cursor is going over right now. There's a green arrow pointing at it. That's called the basal surface. I think, yeah, there it is. I wrote that down there. The basal surface, okay? In this bottom picture here, we're looking at a transverse section of the trachea. So this whole tube here, this whole tube right here is the trachea. Now, how did you know it was a transverse section? Well, you know that when you cut a tube across, you end up with this ring type shape. The space inside the trachea, the space inside any body cavity or organ is called a lumen. So your trachea has a space in it called a lumen. The esophagus, small intestine, they're going to have lumens. Blood vessels, arteries, veins, capillaries, they will all have lumens. All right. Epithelium covering or lining, exposed to a space. Surface next to that space is apical. Surface on the other side of that space is basal. Okay. Let's keep going. A lot of times epithelial cells are really packed tightly together. This too is good for protection, right? If you're covering, if you're a lining, you want protection, so you pack the cells tightly to one another. Down here, this is epithelium from the small intestine. The lumen of the small intestine is up here where I'm writing lumen. And so these cells here, they're all very tightly packed together, which is protective from whatever might be in the lumen of the small intestine. And by the way, in this picture here, if this is the lumen, then the arrow that I'm drawing right now is pointing at the apical surface of this epithelium. Okay, now not only do we pack the cells together tightly, we have to anchor them together as well. I mean, you can't just stand the cells next to each other. You have to attach them to one another. And there are two major types of attachments that we use to bind an epithelial cell to its neighbor. They're called desmosomes. That's one type. And the other type are the tight junctions. What I'm doing here in this top picture is I've got a close-up view of skin cells, okay? Again, I can see a nucleus right here, a nice nucleus. Now, what I want to point out to you in these skin cells, so here's one skin cell right here. Dun, dun, dun. And I don't know, here's another skin cell right here. Let's do this one. In between these skin cells, look at all these little lines. You can see them in between all the cells. These lines are the desmosomes. They're these really strong protein structures that anchor the cells together. Desmosomes are really good when you want the cells to be held together really strongly. Now that makes sense in skin because the job of skin is protection. So you want cells that are packed together with very little space between them, and then you want them anchored to one another tightly. Now, in the bottom picture, I said earlier this is the small intestine. Here is the epithelium of the small intestine with this bracket. I see my nice nuclei of my cells here. Nice purple nuclei there. There's some connective tissue underneath. And as we said earlier, the lumen is right here. Now, what could be in this lumen of the small intestine? In the lumen of the small intestine, there could be food, digestive juices, water. There could be bad stuff too. There could be bacteria, viruses. And what we want to do in the small intestine is we want to make sure that nothing sneaks between the cells. We want to regulate what goes from the lumen through the epithelium into neighboring blood vessels. This is a blood vessel that I ended the arrow in right there. So what we do in the small intestine is we attach the cells together with these structures called tight junctions. And the tight junctions are going to create a barrier between cells that is hard for things to sneak through. That is not very permeable at all. That way we can prevent bad stuff from just going through willy-nilly, and we can regulate what gets through this passage here. All right, epithelial tissue, epithelial cells are lining and covering. They're tightly packed together. They're often anchored together strongly with desmosomes or tightly, almost impermeably, with tight junctions. Okay, let's keep on going. I got some more epithelial tissue right here. Let me draw a circle around a ring of epithelial tissue right there. Actually, I'm not going to do this ring. Let me draw it around this ring right here. I'm just going to draw it in the cells, okay? This is epithelial tissue. I'm drawing a ring kind of through the nuclei of the cells. And here's some more epithelial tissue right there. Notice it has that yellow bracket on it. These rings are things called renal tubules. Renal, R-E-N-A-L, because they're in the kidney. Tubules, because they're little tubes. We'll actually see more of them later on today. And these tubes have been cut transversely. Again, notice the fact that they are rings. And what I want you to see here is there is this pinkish layer underneath them, or around the rings, rather. I got a black arrow on that pink layer right there. Do you see that? Put another couple arrows on that pink layer right there. Right there. Right there. I'm not perfect at ending my arrows, as you can tell. But that is a protein-based layer called the basement membrane. It helps support the epithelium and attaches it to neighboring connective tissue. Now, one thing that's interesting about epithelial tissue is that it is generally avascular. What does that word avascular mean? Vascular refers to blood vessels. The A prefix there means without or lacking. So epithelia are avascular. They are lacking blood vessels. Well, you might be wondering, well, if you're lacking blood vessels, how do you stay alive? Well, there are nearby blood vessels, like this guy right here. It's a blood vessel right here. Lots of them. There's another one right there. And those neighboring blood vessels provide nutrients, remove wastes. There's just no blood vessels within the epithelium itself. All right. Almost done our epithelium introduction. Almost done it. When we're talking about epithelia, we often want to classify them. And there's two basic classifications we can do right off the bat. We can say epithelia are simple if they're composed of a single layer of cells, or stratified if they're composed of more than one layer of cells. If we look at this small intestine over here, this is the lining of the small intestine. We actually looked at this earlier, the lumen's up here. The green bracket is on the epithelium. I want you to notice that I can basically take my cursor here and draw a single line, and it more or less gets through every single nucleus. There's like one or two outliers that got squished away. So all my nuclei are in a single row. That means this epithelium is simple. It's got a single layer of cells. Now over here, this is some epithelium lining the urinary bladder. So the lumen of the urinary bladder with P would be up here. Green bracket represents the epithelium. And look, nucleus, 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 nucleus. There's a whole stack of them. And when there are a stack of cells like this, when there are multiple layers of cells, it is known as a stratified epithelium. Now, of course, which of these two do you think would be better at providing protection? Simple or stratified? Definitely stratified. More layers, more strength. Absolutely stratified will be better for protection. Simple will be better at moving things through, like absorbing nutrients, getting rid of waste, absorbing gases, that sort of thing. Okay, so epithelia can be simple if it's a single layer, can be stratified if it's multiple layers. We can also classify the epithelium based on the shape of the apical cells. So the cells next to the open space. Now, there are three basic shapes. Squamous, which means flat. Cuboidal, which means the cells are roundish or squarish. Or columnar, which means that the cells are rectangular or oval-ish. A lot of times it's helpful to look at the nuclei because the shape of the nuclei will generally mirror the shape of the cell. So, let's look at these. I got a double black arrow here pointing at this epithelial cell right here. Do you see that nucleus right there? Look how flat it is. That nucleus is flat, so this guy right here, the black arrow, is on a squamous cell. Now, what about these guys? Middle picture. These guys, the nuclei are basically circular. So if the nuclei are circular, if you have a circle nucleus, generally the cell will be a beautiful square around it. Not a perfect square, perfect circle there, but you get the idea. And so these guys are going to be cuboidal. That's right, cub. Now lastly, look how ovoid these nuclei are. This cell right here, each of these cells here, is going to be columnar. So, three basic shapes, two basic designations based on the layering. All right. So what we're going to do now is we are going to take a look at some actual tissues in the body, and we're going to start applying these classifications to them as well. Let's do that. What we've got here, this is lung tissue. Notice there's a lot of white space in here, a lot of white space, because you need a lot of space for air in your lungs. The red arrow here is on this skinny epithelium. It is going to get two designations. It is simple squamous epithelium. It is a single layer of flat cells, which makes it ideal for gas exchange. Now, often what happens in the lungs is the simple squamous epithelium forms these little pockets, little cavities, like this little upside down U here. And a cavity in the lung like that is called an alveolus. It is where gas exchange is going to take place. So this is simple squamous epithelium. It is in the lungs. It is a single layer and flat, which is great for gas exchange. It's really easy to get through a very thin layer. It says alveolus here because that's just one. Of course, where we have many, the plural is going to be alveoli. If us is the singular, I is the plural. Okay, here is another slide of lung tissue. I want you to point out simple squamous epithelium. I want you to point out at least one or two alveoli. So pause this if you need to. Point them out. And now we've moved on to a whole nother organ. This is a slide from the kidney. We know the kidney makes urine, makes pee, right? The kidney makes pee by filtering blood. The kidney is constantly filtering blood. And when the kidney filters blood, you actually don't immediately get urine. So you get this precursor to urine that is called filtrate. It's got a great name. Why is it called filtrate? Because it just got filtered. Now, the filtration in the kidney, the filtration of blood in the kidney, will happen at a structure called a glomerulus. It is like a ball-shaped structure. It's a round, spherical structure made primarily of blood vessels. You probably have 1 million of these glomeruli in each kidney. And this is where blood filtration takes place. I can see a lot of red blood cells in here. Okay? Now, Enclosing this ball of blood vessels, this glomerulus, is a structure called the glomerular capsule. My cursor is trying to trace around the entire glomerular capsule. The glomerular capsule basically captures the filtrate. The filtered blood ends up in this lumen, where I got that star, okay? And that's where the filtrate is first found. Now, the glomerular capsule itself, if you look at it, notice it is very skinny. I put 2 arrows on it to highlight its skinniness. It is a single layer of flat cells. So its epithelium type is good old simple squamous. All right, I want you to pause this. See if you can zip back to slide number 6. Find me a glomerulus. Find me a glomerular capsule. Okay? All right. So go ahead and do that and catch up. I'm going to keep on going. Actually, I have a whole nother task for you here. I have a kidney slide again. I want you to pause the video if need be, point out the glomerulus, the glomerular capsule, the lumen of the glomerular capsule, where you, of course, you would find the filtrate, and I want you to also find some simple squamous epithelium. Okay, good. And now, we're going to move on to a new type of epithelium. I've got another section from the kidney. We've actually looked at this picture before. This picture shows me a lot of rings. Look at this ring right here. Ring with a green bracket around it. There's a lumen to that ring. Here's a star and a lumen and a ring of cells around it. Got some arrows and that guy. Each of these guys is a renal tubule. Each of these rings is a renal tubule. A little tube in the kidney. The renal tubules, in their lumens, we would find filtrate, which is the filtered blood that we got at the glomerulus. Okay? So, these renal tubules, of course, they were cut transversely, right? That's why they're rings. And the cells making up the wall of the tube, so here's the lumen, here's the wall of the tube, it is just a single layer of cells. Because look, one row of nuclei. I don't have like rings around rings around rings. And with one row of nuclei, a single layer, it is going to be a simple epithelium. And if I look at these guys, these nuclei are basically circular. So this epithelium is not only simple, it is also cuboidal. It is simple cuboidal epithelium. One of the things that simple cuboidal epithelium is really good at doing is absorbing stuff, like getting stuff to go into the cells. So when you are doing the filtration process to make urine, you filter blood, you make a liquid called filtrate. The filtrate still has a lot of nutrients and good stuff in it. So the filtrate in the lumen of the lumens of these tubes, that filtrate still has nutrients. We need the nutrients to go. The nutrients need to go from being in the lumen to going into the cells. We need the cells to absorb the nutrients. My red arrow here depicts absorption. And simple cuboidal epithelium is really good at doing that. Now, meanwhile, not only can the cells absorb good stuff, they can secrete. They can take stuff out of the cell and add it to the lumen, and that process is called secretion. So the cells can secrete wastes into the filtrate. So as filtrate flows through these little tubes in the kidneys, we absorb good stuff and we secrete bad stuff into it. And in doing so, we turn the filtrate into urine. All right. Excellent. Things are going well here. Here I got another quiz for you. I want you to pause it if need be. Point out renal tubules, find me some simple cuboidal epithelium. And you know what? Just for fun, I also want you to find me a basement membrane in this picture. There's plenty, don't worry. That says basement membrane. Okay, so pause it, find those three things, unless you found them already. If so, awesome. Okay, now we're going to look at another epithelial type. Okay, this is the second to last one we're doing today, so don't worry. This is from the small intestine, all right? The lumen of the small intestine will be right here. And here is the epithelium lining it. My green bracket is on it. Basically a single row of nuclei, oval stretched out nuclei. That tells me that this is simple columnar epithelium. Up here in the lumen of the small intestine, there would be a mixture of food, drink, and digestive juices. And that mixture is this stuff called chyme. I don't want to ever hear you say chime. It's called chyme, okay? Almost every time we have a CH in anatomy class, we pronounce it with a hard K sound. Like 99% of the time, if there's a CH in anatomy class, it is a K sound. So the lumen of the small intestine right here would contain nutrients. We need nutrients to go through, to be absorbed, so they can get into the cell and then eventually get onward into the blood vessel down here. So nutrients need to be absorbed. Meanwhile, wastes need to be secreted the opposite direction. And these simple columnar epithelial cells are really good at absorbing nutrients from the chyme and adding waste to it. Hey, do you remember how we prevent things from sneaking between the cells, like from going in between the two cells? Do you remember what kind of structures anchored the cells together? I'll give you the initials, T and J. Tight junctions. If you said, what are tight junctions? You are correct. Excellent. All right, guys. Let's keep going. I'm zooming in on, well, I'm kind of, hold that thought, need to rewind. I'm zooming in on the right here. Before we zoom in, let's look on the left here, okay? Got another view of the small intestine. Simple columnar epithelium. My cursor is going along the simple columnar epithelium. Lumen up here, which would contain chyme. Now, I wanted to show you this picture in addition to the previous one. Like, I really like the previous one. I think it's a really nice, pretty picture of simple columnar epithelium. But I like this one, too, because I can see this little cell right here. He has a special little cell that was not apparent on the previous slide. That little guy is called a goblet cell. He's called a goblet cell. Goblet, if you didn't know, is like a kind of drinking glass, like a water glass, and someone thought this thing looked like that kind of glass. There's another goblet cell right there. Goblet cells make mucus. And generally speaking, in the human body, there's two kinds of mucus we're going to make. There's sticky mucus and slippery, slimy mucus. The small intestine wants to make some slippery, slimy mucus to make it easier for the chyme and eventually for the feces to slide through the intestines. Okay, we got goblet cells. I also want you to check out the apical surface of the epithelium right here. The apical surface, it's apical because it's next to the lumen. And if you see my blue arrow here, there's kind of like a fuzziness right here. You see that fuzziness? Now, if we zoom in on those fuzzy areas, we see that there are all these like little extensions of the plasma membrane, the cell membrane. If I drew this cell right here, let me draw a cell here. Dun, dun, dun. I know I'm drawing over like all the words. The apical surface has all these little extensions like that. And there's my nucleus down there. Now, all these little extensions What they do is they give these cells more surface area. And more surface area means there can be more proteins involved in that cell membrane for absorption and secretion. By the way, these little extensions here are known as microvilli. One of them, just one, would be a microvillus. Many of them are microvilli. More microvilli, more surface area, better absorption, better secretion. We're actually going to see microvilli in lots of other cells in AMP1 and AMP2. If we want to increase surface area, it is a good bet we're going to have microvilli. Okay. With that, let's mosey on along. Got another area for you, another another tissue slide for you to look at. Pause if need be. Find these 4 items. And folks, we are onward to our last tissue. I think I just have, 2 more slides to go. This one and one more after it. This is our last tissue. What we're looking at here is the trachea. The lumen of the trachea is up here. Here is my epithelium. The green bracket is my epithelium. What I can see here is what do I got? I got nucleus. Nucleus. Do I have layers? It looks like I have layers. But this one is ever so tricky. There is the appearance of layers. But there aren't actually layers. It's just that the cells are like smushed together crazily and their nuclei are at different heights. So it has the appearance of layers, but it ain't actually layered. And for this reason, this epithelium is called pseudo-stratified, falsely layered. Pseudo-stratified columnar epithelium. It has beautiful goblet cells. You can see all these goblet cells. Let's put some red dots in the goblet cells. Beautiful goblet cells. What are the goblet cells making? Do you remember? They're going to make mucus. And do you remember the 2 kinds of mucus we can make? Slippery or sticky. In the intestine, we wanted slippery mucus. What do you think the case is in the trachea? You breathe in through your nose, goes down eventually into your trachea, right? Well, you breathe in lots of stuff. You breathe in viruses all the time. You breathe in particulate matter, particles all the time. The mucus helps trap it. Now, do we want the mucus in your trachea that trapped bacteria, viruses, and particles to just sit there? No, we don't. So what we have are these hair-like extensions here on the apical side of the cell. These guys are called cilia. They're kind of like microvilli, but cilia move. Microvilli just stand there. Cilia basically do the wave kind of upward. They sweep upward, constantly sweeping the mucus up. You are always unconsciously swallowing this mucus. You swallow like a half liter of mucus without realizing it a day. Swallowing it is good because it goes down into your stomach where your stomach acids can deal with whatever nasties were trapped within it. So, this is the trachea. If you see cilia, and the cilia are so apparent, aren't they? The microvilli, I had to like zoom in with my special microscope to see them. But cilia are so apparent. If you see cilia, you're done. You're like trachea, pseudostratified columnar epithelium, and you should know nearby you have goblet cells. All right, folks, we are so close to being done. Last but not least, I have a final quiz for you. I want you to see if you can find me a goblet cell, pseudostratified columnar epithelium, a lumen, and some cilia on this picture right here. Okay. See you next time. Hope this was fun for you. Definitely fun for me. See you later. Bye.